Good afternoon, dear students and colleagues. Welcome back to this module of Research and Publication Ethics, number 3. So last time I had talked to you a little bit about academic and research integrity as well as intellectual honesty. And I had told you that it is desperately needed to have an ethical framework for scientific research, which should be based on moral principles and science in order to promote the advancement of scientific goals. And I also told you about some of these basic moral principles, and I also discussed each one of them. Like we had honesty, carefulness, openness, then freedom and credit, and how to educate or mentor students. Then what is the social responsibility of scientists? What could be the various legal issues which can come in the way? And then opportunities for scientists. Then I talked about mutual respect with colleagues, then efficiency in doing science, and finally respect for the participating subjects, which could be human beings or other living things, or it could be even non-living things like your instruments, okay, through which you get your data and things like that. So I had given you what could be the issues with all these principles. And then I also referred you to this very nice book by David Resnick. Where he gives an introduction to ethics of science. I hope you have been able to go through these reference books, for example, whatever I have shown you so far. And of course, I was expecting more questions, but I think there were only very few questions. So I take it as if that you are able to understand whatever I'm trying to say. And still, I would again say that in case you have any questions on my earlier lectures, please do write either in the chat box, or you can even write to me at my email. Okay, then I also told you about what could be the ethical lash points in the course of a research activity. And of course, I dealt with intellectual honesty and research integrity already. So today, we'll be discussing about the scientific misconducts, which is in short written as FFP. That is, these are fabrication, falsification, and plagiarism, which are considered as one of the most dire scientific misconducts. It doesn't mean these are the only 3 scientific misconducts. There are many more, which we'll be discussing. So today I'll be talking about scientific misconducts, mainly related to fabrication, falsification, and plagiarism. So how do you quantify scientific misconduct? There have been committees which went into this subject, and various committees in various countries, they have come out with certain definitions which can quantify scientific misconduct. So one of the definitions was that scientific misconduct denotes significant and serious deviations from the principles of honesty and integrity and scholarly conduct in science. So, how do you encounter this scientific misconduct? This can occur at any point in an entire research activity, right from the origin of a research proposal onwards, can go through research methodology. Then we have results which come out from certain research methodology that you have adopted, and then analysis of the results, and then finally publication, or either publication of the results in a journal or presentation of the results in a conference. So this scientific misconduct can occur at any of these points in the entire research activity chronology. So now we can ask a question. Since I talked about deviations from principles of honesty and conduct, ethical conduct and all those things. So can we say that there could be a distinct line between what is ethical behavior and what is non-ethical behavior as related to scientific research? Unfortunately, the answer is no. There is no clear-cut demarcation between what is ethical and what is not ethical. So it's a fairly blurred transition from an ethical behavior to a non-ethical behavior. So we can have, on one hand, good science, on the other hand, bad science. But there is a lot in between. Okay. So we can have, if you are really intellectually honest and your integrity is very high, of course, you will undertake good research practices. You will be known for this. And this is an ideal situation. Then slowly we start degrading this, this property. Then we have a certain gray area, okay, which we call as questionable research practices. This is a graph which has been taken from an author, Daniel Fanelli, who has written a number of articles on research integrity and misconduct and things like that. So we have, when you start moving away from good research practices, you can go into an area where there is something called an unconscious bias. First of all, of course, we have some sloppy research methodologies or sloppy research practices. Then after that, we have some kind of unconscious bias in what we do. And then slowly it transforms into what is called a conscious bias in your research practices. And then, of course, start all the serious misconducts like fabrication or falsification and plagiarism, which are clearly, of course, denote very, very serious scientific misconduct. So between the blue and black, I would say there are 50 shades of gray. So now, how do you go from, say, an unintentional error, okay, to a real fraud situation? So here we can see that on the x-axis, you can see it goes from error, misconduct, and at the extreme end we have fraud. While on the y-axis, in the reverse direction, we have the non-intentional or the unintentional practice, and it goes on to a highly intentional practice. So now you can see, for example, wrong observations in the course of your experiment. Okay, so they do not denote a scientific misconduct per se. So they are just simply wrong observations which are purely unintentional on your part. It may, of course, reflect on your inability to distinguish between what is correct and what is incorrect observation. And then we have, once you do have an observation, then if you have a wrong observation to start with, of course, you'll go into a wrong analysis. But you can also go into a wrong analysis, even if you do a right observation, but if you do not analyze the result properly, then that would be called as a wrong analysis. Then slowly we move on to either intentional or the glimmer of misconduct which is beginning to appear. So we have an undeclared conflict of interest. So what do you mean by undeclared conflict of interest? So what it implies is that being a researcher, you may also be involved either in a committee in some capacity, which indirectly has some bearing on the research work you do. Okay, so that would imply, and if you do not inform the necessary, the concerned authorities about your dual role, that you are a member of some committee which can have some interest, so that is called an undeclared conflict of interest. Then we have what is called publication bias. Publication bias has different forms, which I will be talking in detail a little later. But you can, for example, you can think about in publication bias, either you would like to publish in a particular journal which you probably know that it has a slightly lower standards of refereeing, and then in case you feel that your results are not that interesting or that important, a highly reputed journal with a very high impact factor may not entertain your results, then you can go for a slightly lower impact factor journal. So you can have a certain bias in your publication. Then undeserved authorship. This is a very strange issue where you see people often having names in their publications, names of probably quite senior and reputed scientists who may not have anything to do with the research work which you are carrying out. They just happen to be probably your friends or something like that, but still, even though they are not connected with your research work or that particular research problem, you try to put their names just because they happen to be some senior figures in that area of research. So that is a kind of I would say, a misconduct. May not be a very serious misconduct, but it is still a misconduct. Then, of course, we come to the more serious and very serious misconducts where we can have this suppression of data. So this is basically what is called indirectly falsification of data. So you have in your set of results certain data which probably don't conform to your starting hypothesis if you have one. So you try to suppress that particular data which you don't like to present in the open publication or whatever, or a presentation. Then we have plagiarism. It's a very serious misconduct, and which I will try to denote probably an entire lecture to this later. And then falsification of results, which I have already talked about in connection with data. And then fabrication of data. This is also a very serious scientific misconduct where you have a scientific experiment in which you generate some data without even carrying out that experiment itself. So this is pure fabrication of data in keeping in mind what is your research problem and trying to generate certain data which in absence of any actual real experiment itself. It could be even theory also, all right? So without even going into the theoretical examination of a research problem, you simply try to generate data which you feel would get accepted in a journal. So that is what is meant by fabrication. So these are this is how you go from, say, a simple unintentional error or a wrong observation to a more serious scientific misconduct. So why do people indulge in scientific misconduct. So this is again a very interesting thing which one should know. There are various reasons for which this happens. Okay, we'll go into it one by one. Academic pressure and career pressure. Here, usually what happens is that whenever you have an academic position or a certain tenure track position, etc., then you're bound to show certain results as a result of your position in an academic institution. It could be a lot of teaching which is required to be done, simultaneously along with a lot of research work. So this is not easy all the time. So balancing teaching and then research could create a lot of issues, and it can create a lot of pressure because time, as you know, is limited. You have one day to work, and during the day during which you work, you have to balance both these things, where you have, for example, a teaching or an academic responsibility, and then research responsibility. Then you can have personal ambitions and personal biases. For example, you would like to achieve a lot of fame. You want to become a big man in that particular, big man or a woman in that particular area of research. Or you may want to make a lot of money, either through patents or intellectual property rights, for example. So you would like to generate a lot of money by your research and invention, for example, or discovery. And then positions in a certain hierarchical structure. So you would like to go higher and higher in a certain hierarchical structure in terms of your position. So these also could be reasons for driving you to commit scientific misconduct. Then we have another very common situation where it's called publish or perish. Fortunately, this doesn't happen so much in our country, but in many other Western countries, most of the academic positions are tenure-based. And so this tenure is dependent upon how many publications you have generated during a particular period of time. And if you do not happen to generate those certain number of publications, then you may not have a renewal of your tenure at that particular institution. So there's a lot of pressure on scientists to publish their research work to a certain standard which has been imposed on them by the respective academic institutions. So this can also give rise to scientific misconduct. Then we have careless methodology. You have already listened to a series of lectures on how research methodology should be carried out. So suppose you find that if you have to go through all that research methodology, there's no time for you to publish. So you may adopt certain careless research methodologies. Then you have pressure for funding. Again, this particular problem doesn't manifest much in India, but if you go out, especially in the West, this is a very serious issue where you have to keep generating funds for carrying out your research work. So unless you get funds for your research work, you will not be able to go ahead, you will not be able to recruit students, because all these fellowships and scholarships, mostly they emanate by individual research grants which scientists receive. So there's a lot of pressure to be successful in obtaining research grants from various funding agencies. So this also puts a lot of pressure, and you can have a scientific misconduct happening right from a research proposal which you send for funding itself. Then these are certain strange situations like cultural differences. This can happen when people from one culture, which is quite drastically different from another one, they go into that culture or that country. And so sometimes what is considered as normal or all right in your present culture may not be all right at all in a culture where in a different country where you go for work. Then conflicts of interest, which I have already talked to you about, and poor mentoring. So poor mentoring is another issue where when you are PhD students, it is expected that the PhD supervisors properly supervise your work and also mentor students in the right possible manner and also warn them about all these pitfalls which can occur during the course of their careers. So it is partly a responsibility of the research supervisors to mentor the students correctly in scientific conduct and warn them about possibilities of how scientific misconduct can actually happen. So these are various all these properties which I have put in a diagram here. So we have structural factors, organizational factors, individual factors, situational factors, and cultural factors, which all contribute to a scientific misconduct. So you can see that when you have very highly reputed and powerful people, they don't care usually about how you generate the results, etc. So they want more funding, they want more fame and more glory. And that, of course, percolates down to the next person down the line. And finally, the lowest person who gets affected is actually the research student. So now we consider first the most blatant forms of scientific misconduct. So fabrication. So fabrication of data is a very, very bad thing to do. So basically, you don't carry out any experiment. You just by magic or sleight of hand, as you call it, you produce data. So it involves reporting of results of experiments that have never been carried out and also hooking up of data. So fabricated results are never based on actual authentic data. Okay, so you can always have some kind of fabricated data and then build up on that fabricated data and then finally can even present it as a publication. This is actually not a joke, though I show this to you as a joke. This is a very, very serious thing. And many, many scientists have done exactly what is described in this joke. So it is like something like expecting miracles, where you conjure data out of the thin air and then put it in your publications. Then we have another kind of misconduct, which is sometimes related to the first one, that is fabrication. This is falsification of data and results. So here what happens is that your experiments are performed. See, in the first case, in the case of fabrication, the experiments were not performed. But here, the experiments are performed, but the outcome of the experimentation is manipulated. And it is this manipulation of research materials, manipulation of equipment, the processes involved, or modifying or omitting of data, which can result in such a way that the research is not finally accurately represented in the concerned research records. I just show you a very simple example. I don't know if I'm able to take this away. Yes, I hope it goes. Yeah. So you have a set of data points here, which go something like that. Okay. And you find that this is a purely, I'm just showing this to you to illustrate how we can do falsification of data. So you have a set of data points, but then you find during the course of the experiment that there are certain data points which have come out of the general trend. And when I say come out, they are really come out in a very, very drastic manner. So these are called outliers in a certain set of data points. Okay. So, and you can see that because of the presence of these set of data points, okay, you are not getting the fitted curve that you expect, which you should have in normal circumstances should have gone like that. So the temptation is quite high to simply erase this set of data points, okay, and you can see that once those set of data points are gone, you can see that you have a very nice curve which has come out. And that fits exactly with your, probably fits exactly with your supposed, your hypothesis of the research problem. Now, what should you be doing here? Okay, I go back to the earlier slide. Here you can see that whenever you have, I think this was a question which was asked in one of the previous two talks. When you have this kind of data, what should you be doing? See, if there is just, for example, one data point, for example, only this per se, we have only just one data point, probably the fit difference, it may not make that much difference to the fit here. It may still show a fairly reasonable fit to the data points. But then, of course, this particular data point, given this large scatter of, this scatter of data points around this region, it's very clear that this particular data point is bucking this trend and it is clearly out of the trend of the data point. So there could be two reasons. The most common reason is that in the course of the experiment, something has happened at that particular point which caused an erroneous recording of the data point in a different value. That is one possibility. Another possibility is that at that particular point, probably the value of the x-axis, say for example, if you say this is temperature, maybe there was a fall in temperature at that particular instance, and then it went down in temperature. So naturally, the data point should show correctly here. So in principle, the experiment is correct here, but then there was an error of the instrument which caused this kind of a behavior. So then, so it could not record that temperature because it happened so fast that at that temperature which was recorded, it showed a value of the dependent variable which was actually at the lower temperature. That is one possibility. Another possibility that there is some problem with the instrument, and that if there is a problem with the instrument, then this point should appear consistently. Or if there is another kind of a problem instrument, then this point should be closer to the curve, but then of course it should be randomly fluctuating around a mean point over there. So if you find, say, one data point which has come out, it is your responsibility that you should again go back to that that particular point in the experiment, check again whether this particular data point is occurring at the same position or not. Then you have two possibilities. If it is occurring at the same position here, then of course you have to worry about it and see if there is anything wrong with the instrument. If there is something wrong with the instrument, of course you rectify it, and then I'm sure you will get your data point in the right position. But you find everything is fine, and you are getting a repeated occurrence of the data point at this position, then of course there is something, probably something in your sample or the subject or whatever you call it, that is giving you this point. So it may be actually physically true. And just because you want to buck the trend, you're removing that particular data point and plotting the rest of the data point. You might have missed some unique situation in your sample. So this kind of a situation can arise where you erase those data points incorrectly and then plot the rest of the curve. And I also told you about the experiment which was carried out by Robert Millikan, or the famous oil drop experiment. He also removed the outliers in his collection of experiment data points and then presented only those results which conform correctly to his hypothesis. But anyway, that is history. Okay, so then, then we have plagiarism. So this is another very bad scientific misconduct. And what plagiarism involves is appropriation of another person's ideas, processes, results, or words without giving appropriate credit where it is due. It is presentation of someone else's research plan, manuscript, article, or text, or parts thereof as one's own work. This can also involve stealing of another person's ideas. So this also is a very, very serious misconduct which comes under plagiarism. So if you consider fabrication, falsification, and plagiarism, they of course very badly violate publication ethics. Now each of these misconduct, it has a perpetrator, okay, or an offender, and also who is clearly identifiable. In addition, plagiarism, and you see, it also has a very clearly identifiable victim. Okay. So plagiarism, I would say, is more serious because you have two entities, one a perpetrator and also a very clearly identifiable victim. So, and you may ask a question whether this kind of thing happens often. Unfortunately, dear friends, this happens very, very often. It could be happening in your lab, it could be happening in your lab next door also. So it is that prevalent, these kind of scientific misconducts, and it is for your own sake, you must be aware of these kind of misconducts. First principle, please don't indulge in these misconducts. And next, see to it that others don't indulge. And finally, regarding plagiarism, don't fall a prey to it as a victim. So that is the worst thing which can befall a student during the course of his research work. During the course of an employment as an academic faculty or a scientist, if you are a victim of plagiarism, of course, probably you will not lose your job, but then of course you lose your credit for some extent. But if it happens to a student, he in fact loses his research problem itself because someone else has flicked his idea or data or whatever and then published as his own work without giving any credit to the student. So I now go into certain case histories of fabrication, starting with fabrication. Now, I'll first give you the example of this person. These are all, of course, taken from the available records. And I've shown you from where I have taken these case records. There are thousands and thousands of such case records. And in case you people have not gone to such case histories, I really advise you to go through these kind of cases, which will alert you on how this kind of misconduct is carried out. So Bengu Sezen was a lady at Columbia University. She was a PhD student who joined sometime in the year of 2000 or so. Then the investigation showed that there was a massive and sustained effort by Sezen over the course of more than a decade to dope experiments, manipulate, and falsify NMR data and elemental analysis data. And she created fictitious data also. And not only this, she also, of course, created fictitious people and organizations to vouch for the responsibility of her events. So it was a very audacious attempt on her part to do this. So there was a probe which was constituted by the ORI, which is the research integrity body in the United States. So she was found guilty of 21 counts of research misconduct. And the ORI report states that Season falsified, fabricated, and plagiarized also research data in 3 papers and also in her doctoral thesis. And some 6 papers which she co-authored with her research supervisor in Columbia University Chemistry Department, Professor Dalibor Samas, they were withdrawn because these results could not be replicated. So this case began in the year 2000 when this young graduate arrived in Columbia Chemistry Department. And by 2002 itself, there were certain concerns which were raised about the reproducibility of Season's research data. And these concerns were raised by members of the same lab as well as scientists who were outside the university. Now, by the time she received her research PhD degree in chemistry in 2005, her fraudulent activity actually reached a crescendo, according to the reports. Specifically, these reports detail how Season logged into the NMR spectroscopy equipment under the name of some other scientist or some other earlier group member, and then she started doctoring the NMR data, like merging, etc., and then using correcting fluid to create fake spectra which reflected her desired reaction products. So she went to this kind of an extent on this activity. So the reports, they paint a picture of Season as a master of deception, a woman very much at ease with manipulating colleagues and supervisors alike to hide her fraudulent activity. A practiced liar who would defend the integrity of her research results in the face of all evidence to the contrary. Now, Columbia University moved to revoke her PhD in 2010. It took 5 years for them to do this. Worse, the reports document the toll on other young scientists who worked with Season, her lab mates. See, here there was a very serious issue which happened. Because Season was generating data which fitted very well in the research hypothesis of the supervisor of the lab, they were able to publish a lot of papers. So when some concerns were raised against Season by her lab mates, then the research supervisor felt that they were doing this out of jealousy, probably since they were not able to get the same or similar result. And they were trying to put down this lady because she was getting good results. So because of this reason, so the committee found that they wasted time and effort, and the onus of not being able to reproduce that work by their lab mates, it had a very severe negative impact on them. Okay, so at least 3 of those students, among 3 of those students, 2 were asked to leave the lab, and 1 of them decided to leave after her second year. So they realized that there was no point in staying in that lab because the supervisor was unwilling to listen to their concerns. So this is a very serious thing which has happened. So that then the reports also echo sources from inside the same lab. Of course, they spoke under the conditions of anonymity. when this case became public in 2006. So they described Susan as Samus' golden child, a brilliant student favored by a mentor who believed that her intellect and laboratory acumen provoked the envy of others in the research group. That's what I said just earlier. So they said it was hard to avoid the conclusion that Samus retaliated when other members of his group questioned the validity of Susan's work. And she went on to receive another PhD in molecular biology in Germany. So see the wonder this lady has done. So finally, at some point during these investigations, actually Susan vanished. And but her legacy of betrayal, observers say, remains one of the worst cases of scientific fraud ever to happen in the chemistry community. Then we have the case of Ronald Plaster. He was a scientist from Holland, and he was a politician. He was a minister in one of the governments in Holland. Well, that itself should tell you, see, usually you say politicians don't have any ethics. Okay, so the guy could become a politician, so you can see it speaks volumes about his mentality. So here the journal Science, it retracted a 13-year-old paper. Imagine, 13-year-old paper. This was 5 years after one lady, Elizabeth Bick, who was considered as a data sleuth. She first raised questions about the issues with the images in the article. Now, this paper, secondary siRNAs result from unprimed RNA synthesis and form a distinct class. This appeared in 2007. So this was written by a group of researchers in Netherlands and Switzerland. And this senior-most person in this group was Ronald Plaster, who was also the founder of Frame Cancer Therapeutics in Amsterdam, and he was a minister in the Dutch government. And this particular article has been cited 300 times. Okay. So then this paper drew this lady's attention, and in 2015, that means almost 8 years after the paper was published, that she posted her concerns with one of the figures in the paper. Then others also joined in, including to point out the similarities between images in the science paper and two other articles from members of the group. You can see that these are certain what are called slides. People in life sciences know these things much better than me, because I'm not a life sciences person, but I think these are the bread and butter in life sciences, especially those who are working in genetics, etc. So this is a very, very important thing where you have slides of the samples which are published. So you can see that she said the paper appeared to contain duplicated bands in the RNA blots shown in figure 2D. The bottom part of two lanes in two different panels looks remarkably similar. It was not a simple reprobing of the same blot because the marker lanes look very different. Closer inspection of similarities revealed a possible sharp horizontal transition just above the bands of interest, which suggested the bands might have originated somewhere else and might have been copied onto the photo. So this was those things. I think those which have been marked like this, they show the copying of the same slide and passing it off as a different sample. Then she found two additional problems in some supplemental figures, etc., like that. And then again, the lanes appeared to be visible multiple times. So this lady believed that the paper contained inappropriate duplication. So she reported to the journal Science in April 2015, together with 10 other science papers with similar image problems. She asked the journal several times for an update, but then she has not heard back from the journal with an answer about this specific paper. See, this is another problem. A journal like Science or Nature or Cell, they also depend on high-profile papers. And when a question or a concern is raised about a published paper, it also points to the journal and its practices itself. So obviously the referee who was there, who refereed that paper, he missed the fabrication or the misconduct. So it is partly the responsibility of the journal also, because they also become responsible. They try to somehow push it out and they try to avoid taking the responsibility. So it's no wonder that this lady did not get any immediate response from the journal Science. So again, as you said, such lack of action is not unique in our experience of reporting issues to journals. But however, finally, I think in the year 2020 or 19, I guess, she treated a thread of cases of very problematic images where the journal either only did a correction or where they did nothing. So finally, it so happened that the journal forced this Ronald Plaster to retract the paper. But then, of course, they put the responsibility of retraction on the author himself. So the authors of the 2007 science report, so and so, have been made aware of publication or duplications in so and so figures. They attempted to find the original data. So now, what is the redeeming thing for them? They have to find out the original data and show it as evidence. Okay. So they say that they attempted to find the original data, but all the authors, they left that particular institute, Hubra Institute, where the work was performed, and the data could not be located. No wonder. Obviously, if it's going to be fabricated data, it cannot be located. So although the other data in the paper were consistent with the conclusions, all data was taken into account in the review of the paper, and the loss of these figures weakens the conclusions. Therefore, in spirit of full transparency, the authors have decided to retract the paper. So it's a sort of saving grace for the journal, and they have put the total responsibility on the authors themselves. So this particular paper was retracted. So you can also go to this website, Retraction Watch, in case you have not yet seen this. It's a very, very nice site where you have a mind-boggling number of retractions which occur almost on a daily basis. Then we had another case of fabrication by a Japanese, Saizo Miyata. Again, this is taken from Retraction Watch. So this particular scientist, he was a materials researcher at the Tokyo Institute of Technology. And last year, he told Retraction Watch that he found evidence that his postdoc student, Wu Liben, had actually fabricated data. Now, of course, he himself approached Retraction Watch. Okay, that was nice of him. Probably because of his Japanese background, he did this, because Japan, you know that shame is a very undesirable aspect for any Japanese national. So he himself approached the authorities and said that he found evidence that his postdoc student had actually fabricated data. But of course, he did not say who actually uncovered the evidence, but and how. But when he confronted the postdoc student, he confessed. Then he immediately alerted his institute administration and requested the retraction of the paper, preparation of carbon-based catalysts for PEFC cathodes from aromatic polyamide with iron compound. And this appeared in the journal Applied Catalysis A, Journal in 2011. So here the authors have stated that the manuscript contains fictitious data. The scientific community takes a very strong view on this matter, and apologies are offered to readers of the journal that this was not detected during the submission process. That means the referees could not detect the scientific misconduct. So the University Press Office, they sent this journal Retraction Watch a press release in Japanese, but of course they don't want to answer any further questions. The mention of this term fictitious data hints that the Tokyo Tech officials actually concluded that Liben had indeed fabricated data in Miyaka's lab starting in July 2009. So this postdoc was dismissed, and not only he, the university also suspended this researcher, Miyata, and also an associate researcher, which he managed, some person by name Kakimoto, for 3 months without any pay. So Miyata said that he managed the associate researchers such as Kakimoto, who in turn managed the postdocs such as Libin. So look at the strange situation here. He left the day-to-day operations to his underlings. So he made this statement: I didn't know exactly what they were doing in the lab, how they were producing data. Imagine he is the head of the laboratory and he is making this statement. I visit only once a week to meet the scientists. Even though I did not know exactly what happened, I was the head of that project. That is why I had to take the responsibility to resign. Now imagine this very strange situation that being the head of a big laboratory, he is unable to even find out what is actually going on at different corners of the laboratory. So this is an indirect result of having too many students and tackling too many research problems at the same time, and also having a lot of other responsibilities on yourself. So this also can result very unconsciously in a severe scientific misconduct. And since if you happen to be the head of that laboratory, the part of the responsibility will lie with you. Then we have the very famous thing called the Baltimore affair or the Baltimore case. This has been described in Resnick's book. David Baltimore was actually a Nobel Prize winner, and there was a paper which was co-authored by him and was suspected to contain fraudulent data. And this paper actually appeared long ago in 1986 in the journal Cell. Cell is a very, very highly reputed journal in the field of life sciences, cell biology, and it listed 6 authors. So Baltimore, of course, supervised the research, although he actually did not perform the experiment. The paper claimed that experiment showed that the insertion of a foreign gene into a mouse can induce the mouse's genes to produce antibodies mimicking those of the foreign gene. So if this claim were true, it would suggest that one could control the immune system by using foreign genes to make it produce antibodies. It is even relevant to even the present day situation of COVID. So far, this research has not been confirmed by other scientists. And then the experiments were conducted at the Whitehead Institute, as a part of the MIT, the Massachusetts Institute of Technology, and Tufts University. And these were funded by NIH, or the National Institute of Health in the US. Then it so happened there was a postdoc working with Baltimore called Margot O'Toole. And she was under, of course, the supervision of one of the authors, who was Teresa Imanishi-Kari. And O'Toole, she grew suspicious of this research when she found 17 pages of Kari's notes that contradicted the findings of the paper. She tried to repeat the experiment. She failed in an attempt to repeat the experiment and suspected that many of the experiments described in the paper had either not been done or had not yielded results stated in the paper. So she blew the whistle on this research and informed the review boards at the MIT and Tufts. And then the investigation started. So initial investigations, of course, found some errors, but they did not conclude that the research was questionable. And by the time her O'Toole's 1-year term as a postdoc was over, and you can see the situation, after that, she had difficulty finding other postdoc positions for herself, and because she was termed as a troublemaker. But then the ORI, which is Research Integrity Office, they followed up on initial investigations. And then the Congress also learned about this scientific scandal. And then not only the initial investigation, they ordered the Secret Service to step into the investigation. So this went through a lot of things, and even the Secret Service, etc., and a very big scandal. And this committee found that there was indeed a gross, misconduct on a gross scale. And they concluded that probably Imanishi Carey probably put together the notebooks after questions were raised about the research. So in the final report, they concluded in 94 that she had indeed fabricated and falsified experimental data. But after that report was issued, Tufts University, they asked this Imanishi Carey to take leave of absence. But then, of course, the problem did not end there. There was another appeal committee which sat on this problem, and then they exonerated everybody, and everyone simply got off very lightly. But this was the year 1992 and 1995, etc. So much earlier, quite almost 30 years ago, where this kind of misconducts were not so known, and it was also not properly documented how to face this kind of misconduct. So they all almost got off very lightly. Of course, I think Baltimore had to resign from his position as the president of the university, and then they issued a correction of the work in the journal. And then, of course, he also admitted that he did not seek independent verification of the results, which he could have done if he was so sure about his colleagues' innocence. So that was about data fabrication. Now we come to this data falsification. So as I informed earlier, data falsification involves removing outlying data points called outliers from a series of measurements, changing measurement to make it look higher or lower, or reusing the same Western blot. Western blot, I think, is a terminology used in the area of cell sciences or life sciences. So using the same Western blot, either in the same orientation, mirrored, rotated, or stretched to represent a different experiment. Or it can involve misrepresenting results from statistical analysis. Or you can show two overlapping macroscopy images to represent two different experiments. And you can also duplicate some cells on a photo to make it look like a treatment that resulted in a higher cell density. So the first case was that removing an outlier from a series of measurements where you try to knock off data or knock off samples which are not conforming to your expected line of research problem. So this one shows a very interesting patterns where you can see that the ones which are marked, they are supposed to be all different samples. If this is one sample, this is another sample, etc., etc. You can see that exactly the same pattern has been repeated here. So if you are shown, say, this with an untrained eye, you may not be able to detect this fraud here. Okay, and then these 2 points here, you can see here, this one as well as this one. Okay, they're absolutely the same. So they have simply copied the data points and put them here. Okay. So this also can happen, and which is again a very gross misconduct in scientific research. So we again go into some case histories. This was a person of Indian origin at the Duke University School of Medicine. So here there was a problem and concerns were raised, and the Office of Research Integrity, they stepped in, and they found that Poti actually engaged in research misconduct by including false research data in the following research published papers. He submitted the manuscripts, he submitted grant applications, and also the research records. And then the ORI found that he stated in a grant application for funding that 6 out of 33 patients responded positively to a drug, dasatinib, when actually, in reality, only 4 patients were actually enrolled, and none of those 4 patients actually responded to the survey also. And then he said that the 4 CT scans presented in some figure were from lung cancer study when they were not in reality. So he was so audacious to submit this kind of a data in a research grant application. So here he altered the data sets to improve the accuracy of the predictors for response to treatment in a submitted paper. And then in the research record, by doing what? Certain research, this involves some research methodology, reversing the responder status for 24 out of 133 subjects. It's an incorrect methodology for this particular drug predictor in a manuscript submitted to clinical cancer research. Then by switching cancer recurrence phenotype for 46 out of 89 samples to validate certain predictor in a file provided to a colleague in 2008, and then changing certain code values for the cisplatin predictor in a data set provided to another institute in 2010. Okay. So he reported predictors and or their validation by disregarding accepted scientific methodology, so that false data were reported in 9 papers, all in top journals, and they have now been retracted. Then another very, very famous case concerns Jan Hendrik Schoen from the highly reputed Bell Labs. Schoen actually duplicated, falsified, and destroyed data. And he showed a reckless disregard for the sanctity of data in the value system of science. This guy, Schoen, was considered again a golden boy in Bell Labs because he came and started a big storm of publishing papers, almost one per week or something like that. Then he created what are called as field effect transistors, which are the backbone of modern electronics, out of very tiny molecules. And people were in fact presuming that he may be a Nobel Prize material very soon. But then problems started happening. So many scientists who wanted to work on this kind of data, which was put out by Schoen, they were unable to reproduce Schoen's results. And then a certain group of physicists, they noticed that the graphs in 3 unrelated papers, they appeared identical down to what should have been random noise. So what did he do? Schoen substituted whole figures from other papers, removed data points that disagreed with the predictions, and even used mathematical functions in place of real data points. So this was very audacious of him. Okay, so these are the kind of different possibilities where you have fabrication and falsification. And if not serious, you can also have sloppy research. I had shown you one example in my earlier lecture where I had my own paper and which I commented on another work. It was sloppy research work. And unfortunately, that paper was not retracted at all. I'm referring to the result which I showed you in my earlier lecture. And by today's standards of misconduct, that particular paper should have been withdrawn or retracted by the Physical Review B. But this was year 2000, and we were not so much aware of all these issues and that such things could happen. And in net effect, that was not retracted. And it so happens that papers which are erroneous and which are not retracted, they still keep getting citations by future research scientists. So that is one of the very sad part here. So with this, I'll stop at this point. In my next lecture, I'll devote it almost entirely to plagiarism, which is again a very, very important issue, especially concerning in terms of our Indian context. So briefly, what is plagiarism? I have already told you. Plagiarism is the appropriation of another person's ideas, processes, results, or words without giving appropriate credit. And the difference with the other two misconducts is that in plagiarism, there is a very clearly identifiable victim whose credit has been stolen. So thank you very much. I stop here, and so we'll meet again tomorrow when we continue discussing with the issue of plagiarism. Okay. Any questions? Please let me know. Wait. Sometimes in plagiarism check, there are some words marked as plagiarized are the words which are typical. For example, ionic liquid, corol, or hemoglobin. Yes, I agree with this. I'll deal with this in the next lecture. No? No? Okay, thank you very much. See you tomorrow.